Hi, this is José Carbonell from the PerMedCoE project. I'm going to talk about the recently created Center of Excellence that is focusing on personalized medicine. So in this project, we have many different institutions, including Atos as a connection with the industry. In this case, we have some institutions that are more involved in the computational goals that we have in the project, and we also have many others that are more involved in the biological motivations that we have in the field of personalized medicine. So by connecting all these institutions, what we want is to create a strong center of excellence to advance in the research field of personalized medicine in order to provide some cutting edge or some new solutions in order to provide in the future new treatments in a patient-specific way. So this project, PerMedCoE project, have three different pieces. First of all, we want to focus on cell-level simulations. We consider that these simulations are the future of personalized medicine, also the future of many other biomedical paradigms. What we want to do also is to integrate some other core tools, some other essential tools in computational biology that are designed to model molecular pathways. And the idea is to integrate these molecular pathways into the cell-level simulations in order to recapitulate the behavior of individual cells or individual cell types according to what is already now in the databases, according to all the information that we have from the previous experiments that are contained in databases, like for instance, Refactoring or KEK. So in this case, these two pieces that are designed to simulate the biological scenarios that we have in this project, these two pieces are connected into HPC environments because we consider that in order to create realistic simulation, probably we will need to scale up the tools and to use a huge amount of resources in order to, for instance, simulate millions of cells or having hundreds of different molecular pathways. So this is the connection of these three pieces defines the general scope of this project. So based on these three pieces, on the objectives that we have a clear motivation. First of all, the simulation of cellular mechanistic models, like for instance, cell-level simulations are essential for the future paradigms, like for instance, personalized medicine. And we consider that the current performance of these simulators are not still sufficient because these tools are usually fine to work in desktop computers, for instance, or in workstations. And we want to do is to take these tools and to expand their use into a professional HPC environments, like for instance, the Nostrum supercomputer in Barcelona. So based on this motivation, we want to define a sustainable roadmap to scale up a set of essential tools, including cell-level simulation tools and tools that are designed to model molecular pathways, and also to create this entry point to HPC environments, especially to HPC exascale environment, because this is the future of the computation in Europe or in the world. So what we want to do is to adapt these tools to work into these heterogeneous architecture facilities. And finally, having this refactoring of the tools and working very well in this HPC environment, what we want to do, of course, is to translate this omics analysis into actionables that we can apply to daily clinics. So this project have many different steps that we have to fulfill. First of all, is to optimize these tools, cell-level simulation tools, but also the molecular pathway modeling tools, and also to check this and integrate this result into real use cases, into useful use cases to illustrate the benefits of our technologies, and to let the personalized medicine community and other communities related to computational biology research or even biomedicine research, to let them to analyze the benefits of this kind of approaches. Of course, we want to build the sustainability of this project because we want to, in the future, to growth and to have more core tools and to use this kind of methodologies in more different HPC environments, and also in the future to train the biomedical professionals to use this kind of infrastructure that usually is not available, especially for those users that have no special advanced skills in computation. So they are not able to run the tools in a cluster because they don't have the expertise, but we want to provide some interfaces in order to let these kind of communities to use this to create or to do real analysis that could be applied to the daily clinic. So the core tool that we have selected, we have, for instance, PhysiCell. That is a tool that we are using in order to do the cell level simulations, the multi-scale simulations. But we also have 3 different tools in order to model molecular pathways. For instance, we have Cora, that is a nice toolbox of tools in order to model the metabolism. But we also have Selenops, that is very useful to infer and to model signal transduction networks. And also we have Mabos that is able to take some signal transduction networks or even many other networks that are represented in a way of, or represented by using Boolean models, and this tool is able to do like an stochastic models in order to evaluate how are the attractors or how are the steady states of this kind of networks. So by combining these 3 tools, we want to create this initial set of tools to be released to the scientific community. And this refactoring will be based also in the opinion and suggestions of many other experts that we have at the BSc. In this case, we also have many different people that is working already in the POP Center of Excellence. So this is very, very relevant and very important for us in order to detect those specific bottlenecks that we have in the code, in order to properly expand and to properly isolate the tools, and to especially to increase the scalability that these tools have, because in the future we will want us to let these tools to run by using different computers or even by using different HPC environments around the world. So the core tools, as mentioned before, we have PhysiCell, that is a kind of virtual microscope. It's based on physics, and this tool is able to simulate the interactions of the different cells by including physical interactions, but also including the interactions that the cell have by secreting and consuming different substrates that are usually released to the extracellular space. So this tool is based on a diffusion transport solver that is able to evaluate or to quantify the concentration of the different substrates, like for instance the oxygen in a simulation, and we are able to recapitulate some real scenarios, like for instance the necrotic core that we can observe in a tumoroid, or we can also recapitulate how the tumoral cells are able to secrete some growth factors in order to promote the creation of new vascularity. We also have Cora. This is a nice toolbox to analyze the metabolic networks. In Cora, we have many different wrappers in many different programming languages. And usually they implement the most popular analysis, like for instance, the flux balance analysis. We also have, as I mentioned before, Mabos, that is using a continuous time Markov model in order to analyze in a stochastic way the Boolean networks. It uses transition rates in order to expand the pure Boolean networks. And in this case, we can evaluate, for instance, Boolean models that recapitulate, for instance, how is the cell cycle in mammals. We also have Selenops in order to infer, in order to simulate and to model different, by using different flavors to simulate the signal transduction networks. And also we have inside this ecosystem of tools, inside of Selenops, we have Carnival, that is a tool combining other tools, and by using an interlinear programming core, it's able to infer the signal transduction topologies or signal transduction network by using transcriptomic data, for instance, transcriptomic from RNA-seq experiments, or even from single cell RNA-seq experiments. So, but we don't only want to refactor this code tool. Of course, what we want is to combine these tools in real workflows that can describe realistic use cases or useful use cases to be applied in different diseases, like for instance, COVID-19 disease. In this case, in the workflows, the core tools are defined as building blocks. We also have building blocks for other tools or pieces of software that could be connected with the core tools. And the idea is to create like a catalog of different workflows that could be reused, or could be could be released to the scientific community to be used in many different scenarios. In order to do this, we have many different technologies. We are using many different technologies to containerize the different building blocks, and also to create the different workflows by using the standard tools like, for instance, COMPS or Nextflow. So the idea is to create this kind of building blocks. For instance, in a pilot workflow to analyze COVID-19 data. In this case, what we are designing is it's a workflow that combines the single cell analysis of a set of patients and to create like a personalized model by using, first of all, models to select a set of candidate genes that could be interested in the disease to analyze or to evaluate how is the proliferation of those cells that are infected, but also by using physibos, that is a combination of physical models, in order to simulate a set of different cell level simulations in order to predict the different clinical variables of the patient that we are analyzing. So what are the refactoring strategies that we are following? First of all, for instance, we are using Julia. Julia is a nice programming language that in a native way supports distributed and parallel computing. This is the case of Cobrexa. Cobrexa is a Julia package that we are developing, integrating some popular analysis, like for instance, fluid balance analysis or fluid variability analysis that are already integrated in the Cobra toolbox. We also want to analyze and to benchmark different integral linear programming solvers. These solvers are at the core of Selenov and Carnival. In particular, we are interested in Kurobi Optimizator. That is a nice piece of software that could be run in parallel by using different computers. We also are using MPI as a protocol. MPI is a very popular protocol to implement distributed computing. This is a protocol that could be also combined with other protocols, like for instance OpenMP, in order to take advantage of all the resources that a single machine has. It's also very useful not only to distribute the code to have a faster execution, but also to have like a set of different machines behaving like a single large machine. It is something very important in order to face future simulations, including, for instance, millions of cells or hundreds of different molecular pathways. So this is the case of physical MPI. In this case, in the physical strategy that we are following is to take the cellular space and to divide in this space into different subdomains. And the messages that the MPI nodes are passing are related to those cells that are crossing one subdomain to other subdomain. So this is very important in order to keep the coherence and the consistency of the simulations. Also, we are very interested in combining the core tools, in this case, physibos, that is a combination of physical and mabos. And by using this approach, we are able to define the behavior of the individual cell by using Boolean models that are already in the literature and can be analyzed by using models. By using this tool, we can, for instance, simulate the dynamics of different treatments, analyzing, for instance, the proportion of the proportion of tumoral cells that we have in a cell culture, in a virtual culture. But also we can simulate heterogeneous cell populations, including cells that are wild type, combined with other cells that have specific knockouts of some genes that are relevant in the Boolean model in order, for instance, to define how the cell undergoes to apoptosis. In this case, we have different use cases in order to illustrate the benefits of our project. We have a chronic lymphocytic leukemia use case. We also have a tumor evolution use case where we want to use also images in order to evaluate if we can mimic or we can recapitulate the growth of a real tumor by analyzing how are the interaction of the different potential clones that we can have in the same tumor. We also have a use case about the synergy of different drugs. We also included a use case about rare diseases. This is something very important for us. And also, due to the pandemic situation, we included a COVID-19 multi-scale formulation in order to have like a new use case for this situation. In this case, what we were doing is to take advantage of the results that the disease maps community provide us. Also, we integrated the work of Paul MacLean that defined a kind of physical simulation, including not only epithelial cells that have cells, but also including the interaction with other new cells. In this case, the virus is a substrate, so we can evaluate the concentration of the virus around the extracellular space. As I said before, the epithelial cells are the host cells that are infected. We also are able to evaluate the immune cells interaction with the epithelial cells. What we included in the simulation, what we did is to expand the approach of McLean by including Boolean model from the disease map community, from the COVID-19 group of disease maps, that defines how the cells, the epithelial cells, undergo apoptosis when they are infected. So by using this model, we can easily to evaluate how different knockouts produces the apoptosis of those cells that are infected. So this is all from my side. So thank you very much for being here. If you are interested in the project or you have any doubt, please don't hesitate to contact me or to enter in the official website of the project or to interact with us with the social networks that we have. Thank you very much.